Hello, everybody. So I'm going to be giving an update on measurable residual disease detection in CML. And just to remind you why we do this, so there's abundant evidence that the degree to which the bulk of disease is reduced is the most important prognostic indicator for any individual patient. Secondly, a poor primary response or relapse after initial response both suggest the need to check compliance, investigate possible resistance, and potentially change therapy. And finally, we have this new dimension of the possibility of stopping treatments in patients who achieve sustained deep molecular response. So in CML, we have an excellent marker of measurable residual disease in the form of BCR-ABL. So BCR-ABL is essentially a specific marker of the leukemic clone. And we know that quantitative PCR or related techniques such as digital PCR that I'll mention later is the most sensitive and generally available technique to measure MRD. If we look at the BCR-ABL breakpoints in genomic DNA, they're very widely dispersed, particularly within the ABL gene. But after splicing of the primary transcript, most CML patients, at 98%, express either one or both of these two transcripts, termed E13A2 or E14A2, where BCR exon 13 or BCR exon 14 is spliced to ABL exon 2. And these are very convenient targets for qPCR. And many of the tests that are employed around the world use a single set of primers and a single probe that will amplify both of these together. It's important to remember that there are 2% of patients who express rare variant BCR-ABL fusions, and these continue to catch people out, and I'll talk a bit about them towards the end. So this is just a test. It's deceptively simple, but there are some important technical points. First of all, we can do it on peripheral blood, and that's preferred because it's less invasive than bone marrow. We usually ask for 5 to 10 mils in EDTA to arrive in the testing laboratory within 48 hours, and ideally within 28 hours or as soon as possible. And this should be at ambient temperature. Very aware that ambient temperature in different areas of the world may be very different. So it's good to insulate against extremes of cold or warmth. We extract RNA from total leukocytes after red cell lysis, and that's important. If you do mononuclear cells through lymphoprep or any other cell preparation, you will get different results. And then the RNA is copied to cDNA, and there are two core measurements of BCR-ABL, which is estimating the number of leukemia cells, and a reference gene, which is estimating the quality of the sample. And for reference gene around the world, most people use ABL, but GUSB and BCR are also acceptable, and many labs use these alternative reference genes. And then the raw results, if BCR-ABL is detectable, the raw results express as the ratio of BCR-ABL to the reference gene. If BCR-ABL is not detectable, the number of reference gene transcripts is a critical measure of quality for that particular sample. So labs can derive generic sensitivities for their test, and that's okay, but it doesn't tell you the sensitivity for any individual sample. That's where you need the reference gene transcript number. So we have broad recommendations for molecular monitoring. So results should be expressed on the international scale for BCR-ABL measurement. The test should be optimized so it's sensitive enough to be able to detect deep molecular responses. The test should be accredited, or laboratory accredited, depending on what system you're working under. And this means establishing key parameters of the test. So uncertainty of measurement, limit of detection, for example. And then we need to use comparable and standardized definitions of response. So I'll just go through these. This is the international scale. So you have local assays, you may have different reference genes, different bits of kit. And the idea is that basically through the use of laboratory specific conversion factors or the use of calibrated kits or systems, you convert to the international scale, which should be more comparable between centers. And so the international scale is expressed as a percentage, starting at 100% at the top. And very importantly, this 100% is a standardized baseline that was derived in the IRIS trial. It's not the level of disease in each patient prior to treatment. So this is very different to acute leukemia. So in acute leukemia, you look at the level of disease prior to treatment and then at certain time points after treatment and relate the two. In CML, we're relating results to this standardized baseline. So a major molecular response, or now more commonly referred to as MR3, is a 3-log reduction from this standardized baseline. A level of about 1% is roughly the limit of cytogenetics. So a patient who has less than 1% BCR-ABL, you would expect to be in chromosomal remission. And down here, MR4, MR5, this is a 4 or 5 log reduction from the baseline, is referred to collectively as deep molecular response. So we have different methods that labs use to convert to the international scale, and these have evolved over the years. So for many labs, so in Europe, many centers use laboratory developed tests, and then labs need to derive laboratory specific conversion factors, and this is using the methodology developed by Sue Branford many years ago. Originally, this was done by patient sample exchange. More recently in Europe, we've been doing this using calibrated secondary reference reagents that are calibrated to the WHO primary standards. So we developed the WHO standards, freeze-dried cells, about 10 years ago. This is an example of a secondary panel calibrated to those standards. And this is the derivation of a conversion factor process. I haven't got time to go into it now. It works very well, but it is laborious and consequently expensive. The other approach is to use kits and systems that are calibrated to the WHO primary standards, so they directly output results on the international scale. And which of these approaches you go for, they're all equally valid approaches, depending on your budget, your environment, other factors. One of these approaches is more likely to be applicable to your center. This just shows some of the work we've been doing in the UTOS group, using these secondary agents, these freeze-dried mixtures of K562, which is a source of BCR-ABL in HL60. These are available commercially. This was a project which was sponsored by Novartis, and basically it mimics the WHO primary standards. You have target values or levels of disease which approximate to these levels. The exact levels are defined for each batch by reference to the WHO primary standard. And you can use these to measure. This is a Bland-Altman plot here, but this is basically deriving a conversion factor, in this case 0.56, using these standards. And we've found in European centers this works very well. By having commercially available reagents, these are available for labs to do this whenever they, regular basis, when they change equipment, whenever they want, basically. What about kits? There are lots on the system, on the market. Many of these are basically qPCR kits that you then run on your own genetic analyzer. One that really stands out as being different is the GeneXpert system, and this will be talked about in its applicability in a bit more detail than some of the other talks. What's unique about this system is that it goes all the way from blood through to a result. As I said, these other systems are basically, some of them include RNA extraction components, but most of them focus on the qPCR stage. Again, none of these are clearly best. Whichever, different ones will be applicable to whatever you find your situation is. One important point, though, is that using these kits doesn't necessarily guarantee comparable results. Like any complex test, you need to understand its limitations and variability. So we need external quality assurance and internal quality assurance to guarantee quality of results. So we work in the UK and Europe with an agency called UK NEQAS. This is some of the data that they look at. So they send out control samples 2 or 3 times a year. These are measured by labs and results returned. So this color coding just indicates different kits that have all been used, or different kits and approaches, laboratory developed tests as well, to analyze a single sample. And you see there's a pretty big range of results. And even if you're using an identical kit, you can get results that range from less than 1% to more than 20%. So you do need to check the validity of what you're doing. And again, these secondary reference reagents are very helpful in this regard. The other thing is really understanding the basic limitation of the assay. And this is data which I show quite a lot. This was data from Sue Branford's lab, derived many years ago, of comparing identical samples in the reference lab, in this case Adelaide, and a lab that they're interacting with to derive a conversion factor, and then looking at the quality of results. So you see here, as you would expect, there's a very good concordance between the two centers, as is obvious, but if you start making a single point cutoff, so 10% early molecular response, you immediately see the problem. So these samples here are all concordant, they're all above 10% in both centers. These are below 10%, but you have a substantial number of samples that are discordant. So if you're trying to make a clinical decision point, this just indicates the kind of variability you're getting around that 10% early molecular response. And this is shown in more detail here. This is a control sample run on multiple runs, 96 times. The mean value is 11%. And this is the range of results that you get. So you can see from this immediately that it's meaningless to try and decide whether a patient is 12% BCR-able is different from a patient with 8% BCR-able. This is well within the range of uncertainty of the test. So it's very important in communicating between labs and clinicians to communicate that degree of uncertainty. And that uncertainty is more pronounced when you get to lower levels of disease. And that's just an inevitable consequence of the fact that you're sampling small numbers of molecules. So again, this is data from the Adelaide lab. And we were very struck when we did a survey in Europe that people would take their raw results and interpret them differently to derive a final result that was reported to clinicians. So on the basis of this, we came up with consensus recommendations for scoring deep molecular response. Just to remind you, we're talking about these low levels of disease, MR4, MR5, that are particularly used to judge whether a patient might be ready to stop treatment. And also very important when patients stop treatment to monitor whether they are remaining in sustained remission or whether they are showing signs of relapse. So again, not time to go into these guidelines in detail. The paper is freely available, but basically we needed definitions that both considered detectable and undetectable disease. So for example, for MR4, you need to have detectable disease that's less than or equal to 0.1% on the international scale. But if BCR-ABL is undetectable, you need to have at least 10,000 ABLE transcripts in that sample or 25,000 GUS-B transcripts. And correspondingly, lower level of disease that's detectable for MR4.5 and higher numbers of reference gene transcripts. And that's just illustrated in this next slide. So for example, if BCR-ABL is undetectable, you have 45,000 ABLE, that's a good sample, that would then qualify as MR4.5. And just an aside, there's been a fair bit of focus on MR4.5 in discussion points, and that's really because this is about the limit of detection for most samples using standard qPCR assays. So it's not because there's anything clinically particularly special about MR4.5, but that is the sort of target level of disease that you should be achieving in most samples. So if BCR-ABL is undetectable, you only have 14,000 ABLE. It's not a very good quality sample. You can just about exclude MR4. If you've only got 4,000 ABLE, that's a really poor quality sample. Technically, you can exclude MR3. You cannot score deep molecular response. And in fact, most labs would score that as a technical failure. So what about new technologies? How can they help? There's been a lot of interest in digital PCR. So digital PCR is basically the same qPCR test, but it's partitioned into individual either wells or droplets, and then these droplets or wells are then scored as positive or negative in a binary fashion. So you're not so dependent on the amplitude of the PCR, and this means it's a more robust technique. It's less sensitive to inhibition, batch variation, and some technical variance in the assay. And that's illustrated here with a whole lot of different BCR-ABL assays. They're all over the place. You put them on digital PCR and the results are much more comparable. There are other technical advantages. You don't need standard curves, which makes it simpler. Not everyone does it, but you can multiplex the targets of BCR-ABL and the reference gene in the same well, and this gives you an advantage that's less prone to pipetting errors and generally helps to improve the accuracy of the technique. So this improved accuracy of digital PCR has been shown in multiple studies. So if you're interested in homing in on that 10% point or major molecular response, as long as it's optimized, digital PCR is better. Several studies indicating that digital PCR is more sensitive than qPCR. That's certainly true if your qPCR is not optimized, but actually for most studies, the increased sensitivity is pulled out by doing multiple replicates. This is just an example here where you can do multiple replicates and push the sensitivity up to nearly MR 5.5. It is a more expensive technique than qPCR, and the results still need to be converted to the international scale. So it doesn't solve all problems, and there are relatively few labs who are using this routinely for all patients. Certainly on a research basis, or if you're trying to focus in on these deep levels of response, some labs find it very useful. Just to go back to the schematic of the protocol, so digital PCR comes in here. So basically, it more accurately measures what you give it, the cDNA you give it. It does nothing for the quality of the sample, the quality of the RNA, and conversion to cDNA. And these are actually the major variables looking worldwide in the quality of the test. So for that reason, I think there are limitations for the widespread use of digital PCR. So what about atypical B-cerebral fusions? So just to remind you, 2% of CML patients express these atypical fusions that involve the splicing of different BCR exons to ABL. So common ones are E1A2, E13A3, 14A3, E19A2, but there are many others. There are lots of case reports in the literature that these are associated with a good or poor prognosis, but these, I think, are subject to substantial publication bias. Possibly the only exception is P190, E1A2 CML, so that's the isoform normally associated with Philadelphia positive ALL. This has been reported to be associated with a clinical phenotype intermediate between CML and chronic myelomonocytic leukemia. And several cases have been reported with an adverse prognosis. Whether this is to do with P190 or whether this is to do with the fact that many of these patients have additional mutations is not clear. I just wanted to make one point clear because this continues to cause confusion. So if you have a patient with a standard P210, so E14A2, E13A2 fusion, you can detect P190 transcripts in most patients if you look hard enough. If you work it out, these are only present in less than 1 in 100, 1 in 1,000 cells. They're very low level, and they arise either due to alternative splicing or just through to PCR read-through. So this is a genomic fusion, E14A2. This is the major splice, giving rise to E14A2. But it has the capability to produce E1A2 messenger RNA by alternative splicing here. And as I said, with PCR, you can find this. Equally, from the cDNA, these E1A2 qPCRs can amplify very inefficiently a long product here and detect it. So when I'm talking about P190 CML, I mean real high-level P190 with no P210 at diagnosis, not these low levels that are almost certainly clinically completely insignificant. So detecting these rare fusions is very important. I'll just give an example here. 41-year-old female, B-cerebral detected by FISH. She had a good prognosis, low ELTS score, but the B-cerebral transcript type was not determined prior to treatment. At 3 months, B-cerebral was detected at level of MR4. At 6 months, not detected, MR 4.5. So is this what it looks like? An excellent response. So if she had been expressing one of the two major transcripts, yes, indeed, this would be an exceptionally good response to TKI. In fact, she had an E19A2 fusion, which again, in this case, by a long PCR read-through, you can detect at low efficiency and think the levels of disease are much lower than they really are. In the true levels of disease using a specific assay were much higher. And this just illustrates it's important to identify cases with atypical fusions prior to treatment to avoid false negative MRD results. What about MRD detection for these patients? It's difficult. You need bespoke qPCR tests, and we've published bespoke tests recently, part of the UTOS group, along with the development of plasmid calibrators for these. It's probably technically easier to use digital PCR because you don't need standard curves for all of these. These are the kind of tests that should be performed in a limited number of specialist centers because these cases are rare. Importantly, you cannot express these results on the international scale. You can't derive conversion factors for all these rare fusions. But what you can do is look at the sequential levels of disease in relation to a baseline sample, so as is performed for acute leukemias. So what we call individual molecular responses. And you can see clear patterns here of patients who have sustained relatively high levels of disease and those who have what are probably equivalent to deep molecular responses, but you can't precisely define them as such. In terms of the clinical guidelines, the European Leukemia Net is relatively conservative, not recommending stopping treatment for patients with atypical fusions. The NCCN guidelines are rather more broad. They talk about quantifiable transcripts. So this is then just my final slide, summarizing how we get standardized results for molecular monitoring in CML. We want results on the international scale. We need regular monitoring of performance by external and internal quality assurance, and that's if you're using kits or laboratory developed tests. For laboratory developed tests, I didn't talk about this, but a standard plasmid calibrator really helps. And we all need standard criteria for interpreting the results. And I'll stop there. I just want to emphasize that this is really a long-standing international collaborative project to improve molecular monitoring for CML. This started off as research, and some of us, myself and Tim Hughes, started in the Hammersmith Hospital many years ago, and this was instigated by John Goldman. And there are several different strands that have been working together internationally to progress standardization. Thank you very much for listening.